Welcome to the Los Alamos Faith and Science Forum for 2022, our summer seminar series. This season, we started off with our invited external speaker, Professor Marty Hewlett, Professor Emeritus of Cellular Biology from the University of Arizona. He now lives in Taos and teaches at UNM. He reviewed and compared models for human evolution. Tonight, we'll continue to look at that topic, and we will hear about some literature reviews and experiments regarding the origin of life by Patrick Berg. Patrick Berg is a major in the US Air Force. He grew up as the son of an Air Force officer who was always on the move. For college, he attended Brigham Young University. He graduated with a degree in applied physics and also worked for a year and a half in the Williardson biochemistry lab. He went on to medical school at Texas Tech El Paso and graduated with a distinction in research. At the time, he published a number of articles on a post-surgical complication called dumping syndrome, which is about as unsophisticated as it sounds. He completed his general surgery residency at the University of Nevada, Las Vegas, and a fellowship in pediatric surgical critical care at the Johns Hopkins All Children's Hospital. He's a major in the Air Force and clinical assistant professor at UC Davis in the Division of Trauma and Acute Care Surgery. He's married to Lindsay Stevens, and they got married in 2011 and is the father of 3 children. He's a devout Christian who believes that religion and science should be married. Patrick will review some interesting topics on the origin of life this evening. In 1802, William Paley published his famous watch analogy in Natural Theology. He asserted that if a watch is found in an open field, the inference we think is inevitable, that the watch must have had a maker, that there must have existed at some time and at some place or another, an artificer who formed it. He reviewed the complexity of human anatomy as well as animals and plants based on scientific understanding at the time. Seeing complexity far beyond a watch in plants, animals, and human anatomy, he concluded that there is a creator. William Paley's idea was not new. From the Greek philosophers to the Bible and the Quran, the existence of God was asserted to be self-evident on the basis of the wonder of the world we live in. However, after the publication of On the Origin of Species in 1859, scientifically minded individuals increasingly associated the theory of natural selection with the driving force that created the watch. This rigid association fueled an uptick in atheism. However, natural selection as the mechanism of creation falls apart when we get down to the ground level. When we look at the mechanics of what is actually happening in all living organisms, which is biochemistry, the contribution of the theory of natural selection is placed in its proper perspective. It becomes evident that a completely naturalistic view of creation is actually a belief that doesn't come anywhere close to meeting a burden of proof and isn't designed to at any point in the near future. In fact, while sometimes the devil is in the details, this presentation might demonstrate God is also in the details. When the details are examined, the wonder of life is affirmed as well today as when William Paley first published Natural Theology. A cell is the basic unit of all life. Therefore, the natural starting point for exploring whether natural processes explain how life came to be is to explore the development of the first cell. This presentation will briefly explain the primary mechanisms inside all cells, and then review the current degree to which a naturalistic explanation for them seems plausible, based on the current scientific research. Please join me in welcoming Dr. Patrick Berg.
So I've waited a long time to come out here. I wanted to spend time with you guys, get to know you guys. There's a number of people that I've been waiting to meet. Glenn's sitting here right in front of me, so he's the first person I'll mention. Dan, Chick, Nels, Maury. I've spent some time with you guys over Zoom, and I wanted to, talk to you guys in person. But unfortunately, in the process of meeting people and getting to know people a little bit better, I was around somebody with COVID. So that's why I'm keeping myself a little bit distant from you guys, which is a huge shame. But maybe I'll be able to convince the board to let me do a talk next year, maybe, if this isn't a complete wreck. And I'll be able to come out again. All right, so let's get on to the meat of this. So to start, my assumptions for this talk, and also for the intersection between faith and science in general, is that science and religion actually should be married together. Reality, which is often uncovered by science, is at the foundation of a true religion. The best evidence that we have should also guide our thinking. And I've learned that in painful ways in my profession in surgery. And then my other assumption is that whenever possible, using logic is the best way when we're deliberating with each other to come to a consensus about what is true and what isn't. If I use logic to explain my thinking, then I stand the best chance of you all understanding what I'm saying and finding that perhaps your perception in some ways matches my own. The main points of this talk are, first, that life is amazingly complex. Second, origin of life researchers dramatically overstate the significance of their results in a way that is misleading to those without a biochemistry background. And then third, that origin of life researchers have only made progress in synthesizing the basic components that are essential to life in highly contrived conditions. Now, the question is, why am I making these points? What interest do I have in these points? Well, religions throughout history have asserted that a belief in God is reasonable on the basis of the creation. People throughout time have felt that God is real because they felt that the wonder of the world around them and the wonder of their lives can only be explained by a divine creation. The creation is obviously at the foundation of the Christian religion. The Bible starts out with the creation, and through the story of the creation, the nature, the power, who God is, his role in the earth starts to be asserted. And the foundation for a belief in God, this foundation coming from the creation, is also reaffirmed in the New Testament. For example, Paul says that the invisible attributes of God can be clearly perceived by what has been made. Of note, the scriptures of the second largest world religion, Islam, also have passages that rely heavily on the implication that we should believe in God because it's self-evident that he exists because of the creation. And going back to the Greek philosophers, Plato said that everything that becomes or is created must of necessity be created by some cause. But without a cause, nothing can be created. And then he goes on to say that God, being free from jealousy, desired that all things should be as like himself as possible, and that this is, in essence, the truest origin of creation. And I just think that last tidbit is a little interesting because it's reflected in Christian thinking throughout the millennium. Now coming to more recent times, William Paley wrote Natural Theology in 1802. And this is where the watch analogy comes from. The analogy is that if a watch is found in an open field, then there must have been a creator. And this idea became mainstream in the latter part of the Age of Enlightenment. People saw nature as being compatible with or even contributing to a belief in God. What William Paley did in his book is he reviewed the complexity of the anatomy of humans and of animals and plants, and seeing that there was complexity far beyond a watch in these things, he asserted that there must have been a creator. And the word that he kept using is contrived. He said, it looks like these things around us are contrived, that there's parts that serve a purpose and that they're complex. However, this idea obviously took a hit with the advent of Darwinism. So when scientists realized that evolution had happened and that natural selection was an observable reality, this idea of the watch fell out of prominence. Natural selection was asserted as the grand mechanism for creation. Now, a logical question to ask here is, to what degree is there evidence that natural selection is the grand mechanism for creation? Is natural selection, or are natural processes in general, responsible for all of creation? Are they responsible for some of it? Are natural processes the primary driver of creation? Or when we take a step back and look at the big picture and the whole panorama of creation, does it appear that there is not a burden of proof that is being met by the naturalist perspective to say that there is substantial evidence that it all has occurred naturally? So the question that might be asked is, to what degree can you be sure that it all happened naturally? And the answer is that natural selection and naturalist explanations in general, they do not meet the burden of proof. We understand 0.1% of how things in the world around us and how life was created. And so natural selection can't be assumed to be the grand mechanism of creation. Therefore, William Paley's observations are still relevant for today. His points are still relevant for today. To kind of bring in an example, to what degree does natural selection explain the process by which single cells came together about 530 million years ago and developed into complex animals? There isn't evidence that natural selection explains this. Asserting that natural selection explains this is actually a philosophical assertion. But this point that I'm trying to make is actually best made when we look at the origin of life, the first cell. So that's what we're going to do today. And the way to do this, and the way to introduce some rigor into looking at the question of the origin of life, is to look at biology with the lens of chemistry. This is reasonable because chemistry is looking at what is actually going on at the ground level. What is the mechanism by which biology is working? And the natural starting point for trying to determine how life developed is by looking at the starting point, looking at the development of the first cell. Now, people that are familiar with both biology and chemistry know that I'm about to hit a home run because they know that there is a large gap between biology and chemistry in the understanding of the origin of life. And the way that biologists in general have tried to cover up this blatant large gap in their understanding is with something called the prebiotic soup theory. So this is the theory that life started in a prebiotic soup billions of years ago, and that assertion was made without any real understanding of what that actually might entail. It was first asserted about 100 years ago by two guys named Oparin and Haldane. And it was put forward actually with shocking hubris. For example, we have this statement here by Oparin, and he essentially is saying, very, very soon, we're going to understand everything about how life came to be. And this gap between biology and chemistry is just going to crumble away. And looking at that statement now, it looks pretty obtuse. And this is important because proponents of this simplistic view of creation, they, with the wave of a hand, dismiss the wonder of creation and of life, which is the foundation. It is the evidence for God that is available to everybody, regardless of their location on the earth, their culture, their language, or station in life. Now, let's get started at looking at a cell. Cells are so overwhelmingly complex that we actually have to break them down into individual systems to understand them. So the basic components of cells are DNA, RNA, and those two things are the genetic components of the cell, proteins, lipids, and high energy molecules, or molecules that serve as the driving forces, as the gas that makes processes inside a cell go. Now, spoiler alert, we have no idea how any of those components came to be. These components, they function to fulfill the minimal requirements that the first cell must have had. The first cell must have been able to replicate itself. If it couldn't replicate itself, then when it died, that would be the end of it. There'd be no way for the cells to progress, move on. They would have to be able to compartmentalize. They'd have to be able to keep the things that are going on inside the cell away from things outside. Otherwise, the things going on inside the cell would just float away. And they also had to be able to make molecules that served as an energy currency, again, as a gas that drives the processes and the functions of a cell. So the earliest cells would have had to have each of these components. They may not have to have had DNA. And the idea that they didn't have to have DNA is called the RNA first hypothesis. And that simplifies pretty substantially what the first cell may have required. For each of these components, we have no idea how they came to be. Naturalists like to say, well, we'll understand how that'll happen in the future, but that's an ungrounded speculation. We don't know that. And by the best evidence we have, this gap between biology and chemistry is not getting any smaller. Moving on to describing a little bit more of what is inside a cell. Cells are kind of like complex machines. And inside of them, arguably the most important thing is their proteins. And proteins are the things that make a cell work. So if a cell is like a car, then the proteins might be the ignition coil, the alternator, the S-belt, and the struts. For a archetypal cell, there's 2 to 4 million proteins inside it. And this is composed of 2,500 different individual types of proteins. And let me move on. Proteins are made of something called amino acids. So proteins are composed of strands of about 300 units. And these units are amino acids. And these strands fold into configurations that allow the proteins to perform their specific function. And the way that it folds and the way that it's able to perform its specific function is based on the amino acids that make it up. Now, there are an infinite number of amino acids that could be imagined. However, life only uses 20 different types, and I have them here. The simplest type is called glycine, and I'll mention that in this talk a couple of times. And what was thought to be the naturalistic rock of faith was something called the Stanley Miller experiment, which occurred in 1952. So Stanley Miller took 4 basic chemicals, water, methane, ammonia, and hydrogen. He heated them together, and then he fired electrical sparks into his mixture, and voila, he was able to get some amino acids. And since amino acids are the building block of proteins, that was seen as great progress. He reported that he had made 5 different types of amino acids, and 3 of them were amino acids that were seen in life. However, after he died, scientists actually looked at his sealed vials and found that he had made 25 amino acids, and the other 22 were not amino acids that are found in life. So the relevant questions are, how did the amino acids, the 3 amino acids that are seen in life, how did they distinguish themselves, separate themselves from the other 22? Where did the other 17 amino acids come from? And since that time, there have been other experiments that have tried to do the same thing using different conditions. And they've actually succeeded in making a higher complement of amino acids, but no experiment has even been able to make most of the amino acids. And also, all of them are contaminated with many amino acids that are not seen in life. And so those amino acids, they would contaminate any process by which proteins would come to be formed. So those are some of the hangups there, but it's not even the elephant in the room. The elephant in the room is that each amino acid, except for the simplest one, glycine, has a mirror image, and there is only one type of mirror image that is found in life. And this is important because if there was the possibility that proteins could be composed of either mirror image of each amino acid, then the proteins would not fold in the same predictable way. And you couldn't get consistent proteins that were performing the same function. So, spoiler alert, we have no idea how it came to be that all of the amino acids are only one type of mirror image. The mirror image type that they are is called left-handed amino acids. All experiments that have produced amino acids make equal numbers of left and right-handed amino acids. And that's because chemical experiments use millions of molecules. And so it's statistically equal that you would get either type, either right or left-handed amino acids with each individual reaction inside that. All right. One of my objectives in this presentation is also trying to impress upon you that the rhetoric in the field studying the origin of life can't be taken at face value. And I figured that a consensus review might be a good place for me to make this point, because if there's inaccuracies in a consensus review, then it does demonstrate that there's a systemic problem in the field. This consensus review was published in Life. If you read it, it's actually largely a rhetorical piece. It is congratulating a lot of scientists for hypotheses that they haven't actually tested but sound good. Some of those hypotheses could have been tested. And in that case, you can ask the question, well, why didn't they test them? But some of them actually can't be tested. And the reason is because there's just things about the world 4 billion years ago that we're just never going to know. And so I actually think that this paper brings up a fair point, which is how much value is there in speculating about things that we're just not going to figure out. So science is supposed to center on testable hypotheses. So a paper that advances theories that are not testable are by nature not actually science. They're not bad science. They just aren't science. And not even close to all of reality can be framed as a testable hypothesis. So science does not include the whole spectrum of truth. And a lot of this truth is actually fairly important stuff. You have, of course, things that happened 4 billion years ago that we're just not going to be able to figure out. But a couple of things where reality becomes a little more important is, are there logical decisions that we could make, for example, that might ensure that we could have a prosperous future for ourselves? Sure, there are. But I think that we all know what's been said about the best laid plans of mice and men. Sometimes logic fails in this category. For young people, who should you get married to? I think that it would be unwise to approach that decision from a completely scientific mindset. But if the Spock approach works for you guys, then go for that. And then something that historians might ask is, what were people who were living at different parts of the world 2,000 years ago? What were they doing? What were they like? And they may find that in trying to investigate that question, there just is not enough evidence for them to be able to put together a picture of what was going on. And so I think that there's boundaries between science and faith. This quote from the New Testament, I think, is relevant. Faith is the certainty of things hoped for and the proof of things not seen. So faith is centered on hope and on things, it's more centered on things that are not seen. And science is looking more, science is supposed to be looking at things that we can observe. And so there is a boundary there. Even though faith and religion should be consistent with science, we have to admit that there is not always going to be overlap. Somebody wanted me to bring up this question. Am I advancing an intelligent design theory? I am advancing an amalgam of intelligent design and a naturalistic perspective of creation. Is intelligent design science? The answer is no, I don't think it is. Why is that? It's because it can't actually be tested. There were some people that were mentioned by Marty in his presentation that tried to formulate a scientific way to prove that God had created things in the world around us. And this approach not only flies in the face of many central Christian tenets, but it inherently is going to fail. Likewise, my presentation, it utilizes science, but I am not advancing a scientific theory or hypothesis. That being said, science is a subset of logic. And what I am proposing is logical. In my opinion, logic is the most authoritative method of trying to determine what is true. And so I think that it's okay to proceed on that basis. Okay, going back to our review article. So they were trying to determine the different ways in which amino acids, mirror images of amino acids, could be concentrated. And what they said is that there's these mineral surfaces that show a lot of promise in doing this. And I looked at the reference for that statement, and the reference brings you to a review paper that was written in 2010. And the evidence to substantiate their claim in that paper is contained in the statement here that Hayes and et al. First showed chiral selective mineral absorption, specifically of aspartate on common surfaces of calcite. And so if you go back to that paper where the evidence actually is, yes, Hazen tried to use calcite surfaces to select for specific mirror images of amino acids. He claimed that the experiment worked for aspartate. However, if you actually go into the data, he admits that in spite of all of the different experiments that he describes, the best he was able to do was get a chiral excess of 10%. And that's just for one of the amino acids. So what that means is that he was able to get 45% of one type of amino acid and 55% of another type of amino acid. So does this explain how all of life gives you just one type of amino acid? Going back to the original consensus statement, do mineral surfaces really show that much promise in promoting chiral selection? And do they concentrate amino acids? And do they chiral select them? I think that if you subjected that statement to a truth calculator, the output would be false. Now, proteins are synthesized by something called ribosomes. And ribosomes are actually made up of many different types of proteins. And the way it works is that inside the nucleus of a cell, there's a code for all of the proteins in a cell. And something called RNA, which is a molecule like DNA, takes that code to the ribosome, and the ribosome synthesizes the proteins. Now, the question is, how is it that a protein first came into existence when there was no other proteins? Because ribosomes are made up of proteins. So that's not something that has been answered. The way that proteins are made is by the connection between the different amino acids. These connections are called peptide bonds, and they have to link up in specific areas. Going back to our consensus paper, it says that peptide bond formation has been achieved at water-air interfaces under dehydration, hydration conditions, and in mineral interlayers. and that there's been a good advancement in this. Looking at that first assertion, they cited this paper. Going to this paper, there's a whole lot of errors with it. But peptide bonds were only formed in minuscule amounts at the surface of their reactions. And obviously, in order for these oligopeptides, these collections of amino acids to be of any significance, they have to drop down into solution. But the elephant in the room was they didn't actually even use amino acids. They used the ester form of amino acids. And I have a picture here of the difference between the two. So you can see the amino acid on the bottom. It ends with an oxygen and a hydrogen atom. And an ester form has an oxygen atom, and then there's a carbon atom after that, and it can actually continue on in a chain. This totally changes the reactivity of these molecules. And so this study is not progress in any meaningful direction. And there's another group of scientists that actually said studies have explored peptide synthesis at air-water interfaces, and they have had very low yields. I just like this look of Darth Vader. Okay, so who are those other people that made that assertion? They actually have synthesized peptide bonds to, in my opinion, the greatest degree of any of the scientists looking at this question right now. So what they did is they designed something called an abiotic peptide synthesizer. And what this did is this took amino acids in an aqueous solution, and it evaporated all the water, and then it put the water back in, and then it evaporated again. And it would go through these cycles, and it would vary the temperature, the duration of the cycles, the pH, the number of cycles. And doing this, they were able to find a optimal place that they could actually bring amino acids together. So they observed, for instance, that when reactions were run at about 130 degrees Celsius, that they did the best job synthesizing oligopeptides. But this study is a very far cry from understanding how proteins came to be for a number of reasons. First of all, if the reaction was kept at that temperature of 130 degrees for very long, then their yields dropped, and they didn't go into detail on how much they dropped. They ran the reaction at a pH of 9.7, which is much more basic than origin of life reactions are run at. And the other problem is that they only used glycine to good effect. Now, glycine is the most simple amino acid. When they used other amino acids, as you can see, these other amino acids that are much more complicated than glycine, there's other spots that the amino acids can bind together, not the spot that they need to bind together to form proteins. And when they use those other amino acids, indeed, they did bind at those other spots. And in spite of all of this, what is most telling is that they were able to achieve a good result by very specifically changing the parameters of their reaction to get the perfect conditions. And in my opinion, when things are lining up in a perfect way for something to go forward, that's when people think that God is on the move. Moving on to one of the primary functions of proteins, all cells have to have mechanisms of metabolism. And what that means is that they have to be able to create energy to drive the processes in them, and they also have to be able to effectively use that energy. And almost all cells, in order to do this, use a process called glycolysis. There are some cells that don't, but they use analogous pathways. And I have a picture there of the steps of glycolysis, and there's 10 of them. And as you can see, there's complex. Each one is synthesized by a protein. So there's complicated molecules involved here. And what glycolysis does is it takes a sugar and it converts it into something called ATP. And ATP is a molecule that can be used like the gas of a cell to drive the mechanisms in the cell. And we have no idea how ATP came to be or how any of these steps came to be. The other thing is compartmentalization. So, like I said earlier, cells have to be able to keep what's going on inside them away from what's going on in the world around them. And the way that they do that is with something called a lipid bilayer. And lipids are great molecules for that because they have both a hydrophobic and a hydrophilic region. And what happens is that their hydrophobic region will line up with another hydrophobic region of another lipid below it. And it can actually make these bilayers. And the hydrophilic regions will point out to the water inside and outside of the cell. And the question is, well, how did these lipids come to be? The reason that that's a question is because the hydrophobic component is contained of long chains of carbons. And carbons actually they don't naturally bind to each other because a carbon-to-carbon bond is a high-energy bond. That's why lipids are a good source of energy for us. It's because when the body breaks apart those bonds, it releases energy that we can then channel to walk, think, live. Now, going back to our consensus article, it says that there's been recent achievements in figuring out how lipids came to be. But they actually cited a paper from all the way back in 1977. And if you look at the title of this paper, it says Synthesis of Phospholipids and Membranes in Prebiotic Conditions. The paper is only 3 pages long. And the problem is that they took long chains of hydrocarbons that had already been made, and they just combined it with the hydrophilic portion of a lipid. And the question always was, how did those long chains of hydrocarbons come to be? So this title is completely misleading. They did not successfully synthesize phospholipids. And also, by the way, their conditions certainly were not prebiotic. Moving on to the star of the show, DNA and RNA are made up of 4 different types of units. And those units are called nucleotides. And nucleotides are like the letters in a word. And when 3 nucleotides get together, that combination makes a word. And that word will tell a ribosome what amino acid should be used in the protein that the ribosome is forming. And as you can see here by these pictures, nucleotides are fairly complex molecules in and of themselves. I have a picture here of essentially what happens in order for proteins to form. Here you have the DNA that's passing on the code to RNA, which is taking it to the ribosome. And every 3 nucleotides, a new amino acid is being added on. That's an amazing process. Now, nucleotides are brought together by something called phosphodiesterase bonds. And we have no idea how those bonds came to be in the first place. Because right now in every cell, there are proteins that are required to make those phosphodiesterase bonds. And so here is the real chicken and the egg dilemma. What had to have happened in the first cell is there had to have been proteins that could replicate DNA, and the DNA had to have, very serendipitously, actually coded for that protein. And then on top of that, to add an extra layer of complexity, there actually had to have been code in the DNA for other proteins as well that could synthesize that protein to replicate that DNA. If that didn't exist, then after the DNA was replicated and a new cell was made, that new cell would not have the mechanisms to create the protein that could replicate the DNA and cause life to continue. This is kind of a picture of cells breaking apart. It requires coordination of proteins, obviously, to do it. So that's another thing that would have been necessary in the first cell. It would have had to have been able to break apart in a coordinated fashion and share the two different types of DNA that have been synthesized. And now to come to the conclusion of this, there are some biochemists that have asserted a biochemical pathway for the creation of two of the nucleotides in RNA. And when I looked at this study, they begin with glycoaldehyde and cyanamine. Those are their starting reactants. And neither of those reactants are naturally occurring. And so these guys actually wrote a paper in 2015 that went through the steps that they thought could make glycoaldehyde and cyanamine. But I don't want to introduce extra layers of complexity. Just looking at this paper here, how many contrived steps were there to make this RNA? There was 9. They added chemicals serendipitously to their reactions when they needed them. Some of the chemicals were not naturally occurring chemicals. They took chemicals away from the reaction when it needed to be taken away, and that just wouldn't happen. There's no way that the chemical that has to go away is just going to magically disappear. And they used specific pHs to do some of their different reactions. So again, there was 9, at least, being very generous, 9 contrived steps. Another thing to point out is that they actually did not take their starting reactants and run it all the way through this process, keeping the same chemical mixture that they had at the beginning. And nobody does that in this field. So because of all of these different contrived steps, scientific papers are now starting to assert that, well, maybe there wasn't a prebiotic soup. Maybe it's more like there was these streams that were taking these chemicals and serendipitously they met with the right chemicals. And then they would move on and they would perhaps go into ponds or lakes that would then dry up and everything just had to come together perfectly. I actually think that that may have been how it happened, but it seems pretty incredible. And the way that I just described that, it doesn't do justice to the degree to which these reactions are contrived. This guy, Sutherland, he's recently published a paper that says that he's been able to synthesize one of the nucleotides of DNA, but its synthesis is actually way more complicated than the synthesis of those 2 RNA molecules. And like I said at the beginning, the first cell didn't necessarily, it's not thought that it necessarily had to have DNA. It may have just had RNA. So in conclusion, the idea that entropy, which is that things become more disordered, and a naturalistic explanation of the origin life will ever be reconciled at a future point is speculative. What we know about putting together the building blocks of life right now is that it's contrived. This guy, Albert Eschenmoser, he's somebody that was at the head of the origin of life research field. And recently he said that that field is light years away from being able to conceptually and experimentally actually figure this out. So what does this mean? I believe that God had a hand in the creation. And I think that that means that he can interact in our world and he can interact with us. But I also believe that the spiritual transcends the physical, and God's interactions transcend our understanding. And the final point that I want to leave with is that it wasn't the miracles of Jesus that did wonderful things that have blessed our lives, that have eradicated diseases, that have made it so that we can talk with people on opposite sides of the globe without delay, that have provided ways for us to transport ourselves at high speeds. It was people working together with the goal of helping each other, using reason and evidence. And that's what brought about these miracles. So I think, in conclusion, the evidence points to God is an agent in the world, but so are we. And I'll leave with that.